data analysis. And am I the right person to share this with you? I guess yes, because when I started my PhD, so wait, a little bit of background about me. So I did my PhD in neuroscience, so more specifically cellular and systems neuroscience. And I worked with large amounts of data, specifically neuronal recordings, and I had to learn how to code in order to analyze these data sets. And so today I'll be sharing a little bit of my own experience with you, what kind of why I started coding, and then some resources that I used, and I hope you will find it useful. So without further ado, I'm going to share my screen. And if you guys could just comment in the chat about why you're attending this session, and also where you're joining from, that would be great. So again, like I said, today's topic will be learning R and Python for data analysis. And this webinar is sponsored by Editage, and their goal is to make researchers successful, as well as make research consumable. So Editage is the world's foremost academic solutions partner to researchers, journals, publishers, institutions, and corporations worldwide. And again, like I said, their vision and mission is to accelerate global scientific research communication by helping scholars break through the confines of geography and knowledge, bridge the gap between authors and peer-reviewed journal, as well as accelerate the process of publishing high-quality research. They have over 20 years of experience in doing that, and with over 1,300 in-house employees, as well as 2,500 scientific collaborators all over the world. And so they have, over the 20-year period, 20-plus years period, they have helped over 500,000 researchers and executed over 2 million projects in over 190 countries. And it's also been recommended by over 2,000 journals. So thank you, Editage, for making this possible today. So for today's webinar, we'll be going through these 4 sections. First, why should scientists learn coding? So what is the relationship, or why is programming important for scientific data analysis? Then we'll compare the 2 languages we're focusing on today, Python and R, as well as their application in scientific data analysis. Then we'll do demonstration of how to analyze and visualize data using these 2 languages. And last but not least, some resources to get you started. So here you can see the evolution of scientific data. And for the example you see here, I picked genome data. So you can see the Sanger method was developed back in 1977. And over time, there is the Human Genome Project that was announced in 1990, which is around 13 years later. And then fast forward another 10 years where the Human Genome Project was completed. And then there are more advanced methods, right? The 2nd generation sequencer, and then more genetic analyzer, and then the start of the Research Human Microbiome Project. And so why am I showing you the evolution of gene sequencing? That is because you can see as the methods continue to evolve, it actually provides us more and more genomic data. So over time, you start from very little data from just maybe 1 cell, and now you can actually in 2019, it already reached the 3rd stage of the Human Microbiome Project. So you're not just talking about 1 person, right? You're talking about a lot of people. So you now have a lot more data. So what do you do with it? Can you use, for example, an Excel spreadsheet, or something very simple, some more simpler tools that you use to analyze the data? So here you see just a meme, make it a little bit funny and more enjoyable, right? So the teacher say, oh, write, I'm sorry 100 times as a punishment. And as a normal student, I guess you would like really write on paper for that. But as a programmer, you can write the code to execute that for you. So point being, if you learn how to code, then it would be more time efficient, especially in the long run when you process the same data, same type of data over and over again. And also, coding would allow you to extract valuable insights from large data sets. And also, over time, you would be able to tell a meaningful story with all these data sets combined together. And it will also give you more flexibility in generating and customizing graphs and plots to tell your very own scientific story. It is also a universal language that allows scientists from everywhere to collaborate and work together. And last but not least, right, traditionally, most projects actually begin with data collection. But then I also realized that a few years back that there are actually a lot of data sets available that can be repurposed and used to answer your specific scientific questions. And I'm guessing there are also a lot of PG projects out there that simply requires you to analyze existing data. So here are some examples of data analysis and visualization with Python, right? So you can be looking at crop yields using a line plot, or restaurant bills in a bar chart, right? And then here, looking at sepal length and width in flowers using a scatter plot, or looking at flight traffic using a heat map, and checking the distribution of sepal width with a histogram, and many different possibilities out there. So now let's compare the two languages, Python and R, as well as their applications in scientific data analysis. So Python, I will go through this, compare these two languages in these categories. So we'll look at the objective, why this language was developed, and the workability of this language, integration, database handling capacity, as well as the environment that you can execute them in, and some essential packages and library, as well as advantages. So starting with Python, Python is typically used by data scientists or for web development or embedded systems. So it can easily perform a matrix computation as well as optimization. So it programs, they're integrated with web app for easy development. And it can also handle large amounts of data very easily. And you can execute this language in many different environments, including Jupyter Notebook, which I'll do the demonstration in today. And there's also the Spider IDE, which is designed by and for scientists, engineers, and data analysis, as well as IPython. Some essential packages and libraries include NumPy and Pandas, and these two packages allow you to manipulate the data set. For example, you have a CSV file with all your data in it. So these two packages will allow you to manipulate the data, for example, do some calculations, create a new column based on these other columns, right? And then you have SciPy, scikit-learn, and TensorFlow that do more statistics and machine learning, or even deep learning applications. And advantages of using Python is that it has a lot of functionalities that actually go beyond data analysis and statistical modeling. Like I said earlier, you can do a lot of machine learning and deep learning with it as well. And it is also easy to learn. And so on the other hand, for R, it is actually built for data analysis and statistical modeling. It also consists of many easy to use packages. It typically locally run programs, and so it poses problem for handling larger data sets. And some some environment that you can execute R in include RStudio as well as R GUI. So I'll also be showing you some free resources offered by RStudio that allows you to do a lot of things with R. And some essential packages like ggplot2, Tidyverse, and Caret. And the advantage is that it produces quality plots with users having full control over the design. So you can see like R might be a more scientific language compared to Python, but then both can be used depending, both can be used, and it really depends on your use case and what you're looking for. Typically, Python is a bit easier to learn and start with. So Python is actually a more general purpose programming language, right? That can go from web application or program development to data science. And you can see here on the right, some example plots that can be generated using Python libraries. So Python is, like I said earlier, easier to learn. And it also contains many libraries for machine learning and deep learning, like TensorFlow, PyTorch, and scikit-learn. So it depends, again, on your use case. These libraries can be explored to help you in your scientific research. And some useful packages in terms of data analysis and visualization include NumPy, Pandas, Matplotlib, and Seaborn. So I have included the links to all these, so you could also explore on your own. And typically, for each of these libraries, there's also documentation and some resources to help you get started. So R, so I'm gonna play this video here because you can see you can also generate interactive plots. In Python, you can also do the same, but I guess this is from one of the R libraries. So R is, again, specifically for statistic computing and graphical computation. It continuously growing, right, with thousands of packages that can be used for many, many different applications. So some of the useful packages are Janitor and Data Validator, and Deploy, and also ggplot2 and Plotly. So these packages allow you, again, to manipulate and clean the data, and then also prepare them for data visualization. So now would be a good time to show you how to perform data analysis and visualization using these two languages. So let me just switch to So just want to emphasize that this demonstration is generated using the IRIS-Flower dataset that is free and publicly available at the UCI Machine Learning Repository. And so, but then this dataset has also been uploaded to Kaggle, which is a platform that a lot of data scientists use. And Kaggle contains a lot of data sets that you can easily create a notebook and explore, which I'll also show you in a minute. And the data set can then be downloaded from there, either locally to your device, or you can also explore the data set on Kaggle by having a Jupyter notebook there. So just a little bit about this data set. The IRIS-Flower data set is a multivariate data set introduced by the British statistician and biologist Ronald Fisher in his 1936 paper. And so this data set contains 150 records, and it consists of 50 samples from each of the 3 species. Right? There's the Setosa, Virginica, and also Versicolor. And 4 features were measured from each sample, including the length and the width of the sepals, and also the petals in centimeters. So to generate this Python notebook, the code is actually adapted from another notebook. And you can also explore later. So that notebook, wait, let me see if I can show you quickly in a bit. So first of all, when it comes to programming, like I said earlier, right, for both Python and R, there are many libraries and packages that has already been developed to perform different tasks. So the first task you do here is to import these libraries. And so, like I mentioned earlier, NumPy and Pandas, right, that you can use to manipulate the data set, and for data analysis purposes. So when you hear say like import the package SNP, it means that when you run the function, let me see if I can find an example here. Or sorry, I'll use pandas as an expression, right? So you can see the PD, right? We imported pandas as PD. And so you can see if you want to read a function from this library, then you would start with PD dot. So it would be what you imported the package as, right? And then you add a dot, and then it will be the function that you would want to run. So let's try this. So now I'm going to run this function, right? So import all the libraries. And then to access the data, I have it uploaded to my Google Drive, so I would also need to do so in order to give access. And then once you give access, and then you can read in the data, right? So I named the data Iris because it's called the Iris flower data set. And then you can say reading this CSV file from, this is the path where the file is saved in. And then you can say, okay, I want to know what's contained in this, like what are the columns contained? Because I mentioned earlier, there are four features measured, right? So you can run this and you can say, so one good thing about using Google Colab or like Jupyter Notebook is that you can see the outcome, like the output right below. And so, I wanted to see other column names. So here you can see it's sepal length, width, petal length, petal width, and that's also species. And so now I say, okay, can you print 5 random rows of data from this data set? And see here you can see the index of each row, and also like the data for each feature that was measured. So now we know how this data set looks like. And here for demonstration purposes, we're actually not going to do too much data manipulation, but also just to show you how you can generate plots with the data set. Oh, sorry, it should be. And so, you import the Matplotlib is one of the libraries that you can use to generate visualizations. So here, first you create like subplots, and then you can scatter. Here the goal is to generate a scatter plot, and so you can have the function scatter here, and then other functions that allow you to add more information to your plot, like setting the title as Iris dataset, and then the x-axis label to sepal length, and the y-axis label to sepal width. So when you run this function, it will then generate a plot that looks like this, right? So again, if you want to like remove this underscore, you can also do that, right, by manipulating it here and simply say run. And you can see now everything is removed, right? And you see a scatter plot with both the data from the two columns. But it's not helpful here, right? Because everything is blue and you don't see where the data is coming from, which column it's coming from. So then you create a color dictionary, right? You specify the colors, right? So for Setosa, it's going to be red. For First color, it's going to be green. And for Virginica, it's going to be blue. And then you do the same thing again. So now, again, you can set the title to iris data set, and then I would like to remove these underscores here. And when you run this cell now, and you will see now you can generate or you can tell which species, the data of each species contained in this scatter plot. So this is one library you can use, Matplotlib, and earlier I also mentioned you can use Seaborn. So very similarly, again, we imported Seaborn as SNS, right? So this is the package name, and then this is the function within the package. And so you specify what you want the x-axis to be, what the y-axis to be, and where you should be, what data it's getting, what data you plotting, right? So the data would be iris. And so you run this again, right? Again, you would get a scatter plot with uniform color plots. So again, this is not helpful, right? We want to differentiate them. So what you do is that you tell the function that you want each species to have a different color, right? So here in Seaborn, you do it by hue. And then when you run this, it would generate then using the preset default colors, right? One color for each species, again, allowing you to differentiate what is being shown on this plot. So that scatter plot, right, to visualize the relationship between these two variables in the three species. And so, if you want to visualize the distribution of data, then you can use histograms, right? So using Matplotlib, you can simply say iris.plot.hist, right? So and it would if you want to, so here the goal is to generate so we have 4 features, right? We have the sepal length and width, and also the petal length and width. So the goal here is to generate 4 histograms. So you can do that by specifying you want subplots, right? More than just one plot within your plot. And you would like the layout to be, 2 rows and 2 columns. So it's like a 2 by 2 plots you generate. And you can also specialize the figure size, right? And also the number of bins. And here, when you run this, right? So you can, this also follows the default color scheme, right? So you have the sepal length with 20 bins, right? And you can see that it actually most of the data belongs to more the far end on the right side bins, so the bigger values, right? And then you can see that they all share the same x-axis, right? But different y-axis depending on the frequency. And so this is one way of just exploring the data, right? Showing the distribution. Then if you want to visualize the correlation between each variable, you can generate a heat map. And to do that, you first need to generate a correlation matrix before you can do the heat map. So essentially, the matrix contains all the values that the heat map should display. And so I'll just quickly show you how it would look. Oh, there's an error here. Let's see. So when you run into an error, so it would tell you what the error is. So it would tell you like, okay, the module matplotlib.pyplot has no attribute pyplot. So this means that function I'm calling, or like the feature I'm calling, is not part of this function. So what I need to do is to remove this, delete, and that should run, right? Because it depends on how you import the libraries. And if you say, if I already specify that pyplot is part of, when you import it, the short form already contains that, then it means that I'm already running that function. So what I did essentially earlier is that I ran So PLT already is stand for matplotlib.pyplot. So essentially what I did was running pyplot twice. So this is not a module within pyplot, right? So what I wanted to call is actually subplots. So I'm like running this thing twice. So I need to remove it, right? And then now it generates a heat map with all the different values and also, the what each cell corresponds to, right? So when you compare like sepal length with sepal length, then you always get a 1, right? Because it's against itself. And then if you want to compare or look at the correlation between sepal length and sepal width, then you can see there's like a slightly negative correlation. So you can also do the same thing, generate heat map using Seaborn. So again, we import a Seaborn as SNS, right? And the function you're calling is heat map. And we already generated the correlation matrix. So you just call like iris.core, so that generates the correlation matrix. And then and not true means like you want it, you want the numbers to be there, like so you want the graph to be annotated. So again, you can see all this information here. I personally like using Seaborn a little bit more simply because graphs generated or like the default color schemes look a bit nicer than Matplotlib. And but again, this is personal preference, and like most of the things you can actually change, right? You can specify like what color you would like. Yeah, so these are just some examples of what you can do to visualize data in Python. And if you want to visualize the relationship between each feature, you can also do that. So typically, if you run the function pair plot within Seaborn, you would get both histograms and scatter plot. So depending on what you're trying to show and visualize here. So if you have any questions, don't hesitate to ask in the chat or in the Q&A box. And so we'll get to them later. And so this is what you can do with Python. And you can also do something similar in R and with the same data set, which I will show you just in a few seconds. So I want to show you that Jupyter Notebook or Google Colab, more specifically like for Google Colab, they actually focus on supporting Python and its ecosystem of third-party tools. So if you want to run R code in Google Colab, you would have to run a few functions. And for each cell that contains R code, you will need to tell Google Colab that, hey, I want to run R code here. And then you would need to use, just type this in here, right? 2% signs with an R, and then it would tell Google Colab that like, hey, this is not Python and this is actually R. And then you can run this all the R code then. So for the purpose of demonstration, I will show you Kaggle as well. And so now, right. So Kaggle is, so this is how it would look like if you, after you've selected a data set. And so you need to create an account in order to use it. And so this notebook, it's been uploaded, it's been up here for 6 years, right? You can also see like how many people have viewed it and how many people have upvoted it, like, they liked the notebook. So this person, like Antonio here, he visualized the iris data set using R. So you can see under the notebook tab the commands, right? So everything here is a comment, right? So this here you can already directly run everything, unlike in Google Colab. So it's specified that like, okay, I want an R environment, so you can execute these codes. And so it loads libraries a bit differently. So it might be more intuitive here, right? Instead of importing a library, and here you say like library, and then the name of the library. So ggplot2 is, or ggplot is very popular in terms of like a lot of people, when they use R, they use this library ggplot2 to visualize the data. And then there's also like other libraries used here to, sorry, you know, depending on like, so to either read in the file or generate the other plots that you would see here. So again, here, using the readr library, right? So you can read the CSV file. And then, so here, if you want to look at the first 10 rows, right? Just random 10 rows of the data, then you would run this here, right? Sample and the number of row, which data set, right? From IRIS at 10. And so again, you would see, you know, just a random collection of them. So there are multiple ways you can visualize just parts of the data. You know, sampling would be one way, right? Random sampling. I also prefer using random sampling because sometimes, like, you know, the information might just look right in the first, I don't know, 10 rows, and it might not in the rest of the data set. So I always find it a bit better to just, like, you know, look at, like, just randomly sample and look at the overall data set. So you can also run functions like, for example, in Python, like head. So just look at the top, I don't know, 5 to 10 columns that you would like. And then here, you can, again, look at generate or, you know, investigate the density and frequency using histogram. And so this is the function you will be using, ggplot. And if you visit the R, like in the documentation for this, let me show you. Maybe I can quickly show you how it would look like. Right. So they typically have cheat sheets that you could use for data visualization. Right. I'm just gonna quickly share this with you. Right. So if you want to visualize data with ggplot2, you know, it will show you what function you need, and also typically how it works, right? So you first give the data and then maybe the function and how it's being mapped. And things like here, it would specify what you need to, like, it shows you here what you need to specify for the function to work. And so going back to what I was sharing earlier, or do you still see, sorry. Right, sorry. So here, right, again, for each feature, you would have to specify what you want to show, right? So again, but then instead of earlier in Python, we saw like different lines, and R is typically done in one single line where you have the function and then all the variables is contained within that. And you do that, right, for length, width, and also petal length and petal width, and then you combine them together, right? For each, so this for each feature, right? And then you would generate a plot that looks like this, right? So each color represents each species, right? And then for the 4 features, it looks pretty neat, right? When you generate plots with R. And so you could see, well, we could see here, most of them follow a normal distribution pattern, right? And so we could also see that the measurements of very small flowers in the petal width and length column. So we can then review the density distribution of each attribute broken down by class value. So we can use the density plot here, right, to help see the separation between the different classes, right? Also help us to understand the overlap of class values for an attribute. So again, you use ggplot, right? So here you specify how each feature should look like, right? So again, there will be 4 features, right? One for petal length, one for petal width, one for sepal width, and one for sepal length. And so here again, you arrange right? Earlier we saw it's like a 2x2 plot, right? So it's the same here. And so you can see the density density of these values, right, for each species and for each feature. And so now, when you use a box plot, right, you'll be able to identify outliers that falls outside of the typical range, right? So you can see that actually some classes do not overlap at all, especially in petal length, right? You can see they have three separate, like the values are three separate clusters here. And so what you do is, again, run the function, right? You specify what you want to see and what the color scheme should be based on, right? And then you can generate box plots. And so here, each dot represent an outlier. So here you have the median, you have the 25th percentile, 75th percentile, and everything that falls outside of that range. And so using box plot allows you to see these outliers, right? And so now here, right, to visualize all the variables within one single plot or one single visualization, right, that contain all the box plots. So again, for each feature you want to show, you specify how it should look like. So in our this symbol here means equal to. So it's like this is a name of the variable equals to this here. And so again, you use grid, right? This is why the library was imported earlier, right? To arrange the plots so then you can see everything at once, right? So you can see all four plots in just one bigger plot. So like 4 subplots in one bigger plot. And I'll just quickly show you what you can generate with them. So here in this case is to visualizing the data using Violin plots which then allow you to look at the distribution as well as identifying outliers, right? They're very similar to box plots, right? They show the number of points at a particular value by the width of their shapes. So if it's like slightly fatter at the bottom, then there are more values at that range. So you can see again here, right? So it's like you can see the Violin plot, and also you can see the distribution, right? You have the median, again, the 25th percentile, 70th percentile, but then now Violin plots actually contain a bit more information, right? It also shows you how many data points fall into this range, denoted by the shape. So here you can see most of the values actually fall in the median for the Setosa in sepal length. So I'll share the links with you later. Like you can explore all these notebooks on your own. It's very fun to just run the code and see what it generates, and then you can change things here and there and see the results. So yeah, so this is like just wanted to give you an overview of the types of plot that you can generate, as well as how the code will roughly look like. And of course, like, find any data set that interests you, perhaps like something that's already been cleaned and you can directly visualize, and play around with all these functions, for both Python and R to see, just to get a taste of what it's like to visualize the data using these functions. So now going back to the slides. Wait. Right. So after showing you how to visualize the data with these two languages, I want to briefly talk about version control and why you need it. So I can speak from experience that at one point I didn't use it when I was in the lab, and we had a ransomware attack where I actually lost quite a bit of my code. But then, it happened already to a colleague of mine, and where he actually lost everything. And so we started like, backing up all our codes. And it's important because, it took a lot of time to write these codes, and you don't want them to just be lost, right? So to, protect yourself and also to protect your project, use version control, and I'll tell you now why. There are more reasons than to not lose data during a ransomware attack. So in general, so version control is also known as source control or revision control. It actually allows you to track and manage changes to code or files in general. And especially if you work in a team environment, this is very important, right? So some advantages include like prevents data loss, right? For example, from ransomware attacks. And it also allows better collaboration, right? What does that mean? Multiple people can work on the same project or document at the same time, but it won't create any conflict. I'll show you in the next slide how it looks like. Collaborators, right? So different people, they can commit and merge changes without conflict. They can also edit shared documents without unknowingly overwriting each other's work. So you can see here, right? You have the document, and then you have like, one person working on some features, and then another collaborator working on something else. So what happens is that, like, with this very original version, right, you created a sub branch of it. That's and then it would merge back to the main branch and contribute to the document that you were editing earlier. And then for this collaborator, he would then, grab a version of what's going on already, right, and then contribute to it, and then merge back to the original, the source file. Not source file, sorry, like merge back to the version or like the document that was being edited earlier. And so this is like the version that now all collaborators can download, or like they pull the changes, right? So any change that's been made to this starting point here, right? You can pull all the changes from this version. So everyone, no matter where you're working from, no matter who you're working with, everyone will have the same updated code when they pull the changes that's updated to this version. So Git is an open source version control system. And they actually think of data as part of a bigger project, right? So here I will show you. Git actually takes a picture of what all your files look like at that moment. So for example, and then it stores a reference to that snapshot, even though you didn't make any changes at that point to that document. So then you have version 1, file A, B, and C. And say, I worked on file A, and then you worked on file C1 or C. And then it generated a version like A1 and C1, but then nobody touched file B. So instead of just leaving a blank here, right, it automatically incorporates what file B would look like, and then you have a complete version here, right? That allows you to easier access and manage your data, because you can just pull this version and you would get everything that you need for that particular project at that very particular moment. So, I know this class is about like learning R and Python, but I think knowing about version control very early on when you start coding is very important, right? Because it allows you if you made a mistake in your code, you're like, oh, you deleted a huge section that, and you can always go back to your previous version, right? And you can say, oh, I want this previous version now. And then you can simply pull the difference, right? Pull the changes from that version to your current code, and then, nothing is lost, and you can still even see, what's been made, what changes you've made at different stages, and then you can reverse that change if you know that it somehow broke your code. And so here I'm gonna show you some resources on how to get started, right? So like Kaggle, I mentioned earlier, there are, they have courses on like, introduction to programming, Python, right? And then Pandas specific package of Python that's very useful. And then there's also something on data visualizations and welcome to data visualization. And once, if you are planning to learn Python, once you have some basic knowledge, then you can also go on FreeCodeCamp, and they have a course on data analysis with Python. And I like it because, let me show you, because they also, so this is how the course would look like, right? And then what you will be learning, so introduction to data analysis, how you do it, and then how to use Jupyter Notebooks for your data analysis, and then talk about the different packages used for data analysis. For example, here, NumPy, Pandas, and then how to clean data using these packages, and then reading different file formats, right? And then parsing data and also saving data, and generating Python functions. And so there are lots of information here that you could use that are very helpful to begin with. And so like then you have some Python projects as well that you have to do that actually allows you to practice what you've learned. And this is one thing why I like the course. And so there's also for R, there's Post-it Cloud, which is actually share this. So here it will show you the many different resources that you could use, right? Some basics of using R, and then also how to transform tables using R. So you can just like click on them, or like visualizing data, for example, with a scatter plot. And so it will show you the different steps, right? And then the function and what you need to put in it, right? So here is very useful. Like, okay, you need the data, and then the mapping looks like this, right? X is column A, Y refers to column B, and then for each point, or like for the color scheme, based on column C. So there are like a lot of examples that you can look at, and then you can see when you run this code, how it would look like. And then there's also like from Geeks for Geeks, data visualization in R. And then there's also this book that's I think available on GitHub, right? How to do modern data visualization with R. And then if you want to learn more about Git and GitHub, you can also go to their website. And yeah, so the two notebooks I used in this session would be the Introduction to 2D and 3D Data Visualization, which is also available, which is also on Kaggle, and then also the Iris Data Visualization with R that I just went through parts of it with you earlier today. So these are some resources to get you started. And once you feel comfortable, or you can even maybe always start with one language, right? You can learn Python first, and then maybe switch to R. And depends on what you're comfortable with, and also depends on what your research or like what your lab uses, right? And so now would be time to ask some questions about the learning process or learning journey. Yeah, so thank you all for being here. So I will answer questions now. Right, so one question is it possible to import all libraries you will use for some time, or you import a library when you need it? So this really depends on you. You can do both, actually. I personally prefer importing all the libraries at the very top, just like, for keeping track of things. And also, it's like a better practice. Like, you import all the libraries at once at the very top, and then you would see what you already installed, or like what you have already imported for this particular notebook. Okay, and then in terms of, wait, can we input our own data in R? Yes, of course. So there was a function I showed earlier for R when you import your own data. So you just need to specify the path where your own data is stored in. Wait 1 second. Okay, I can share this with you. Right, so here you can load your own data set. using like read CSV, or depending on like what file format you have. And similarly, in Python, it was also the same thing, right? We also read in the file here, right? You specify, like for me, I had to specify first the path, right? So this is the path where I stored the data set. So it would be similar in R, but you just use a different function. So here I use pandas read CSV, and for R, it was just read CSV. So it was from this library, right? Read R, and then this function here. And now another question. So could you copy a code and you use it in another project, or do you request to write it? So like what I did here earlier, right? I also showed you examples of other people's code. So I always give you the reference where you could see it. And it also really depends. So there are a lot of open source projects that the code is made available where people can just contribute to. And I think it would also always be right, to credit the source. But then there are also a lot of codes that are just common to public. If you, for example, if you wanna plot, like generate a scatter plot, this is like a pretty standard function. And so it really depends on like, what codes you're copying. But it's always good to start with like known codes. So for example, I showed you in the free resources, right? So there's a book called Learn R Through Examples. Okay, let me share with you. And so here it really, so this is like, you can access it. It's on this person's website. And so it even walks you through how to use RStudio, which is one platform or one environment that you can run R. And so, here you can just, it really walks you through. So what I recommend doing is like having RStudio open and then this book on the other screen. So, and then you just type in the code that you see here. This is like the best way to learn and experiment with a language. And you can, is it possible to use two data sets and import them in R? Yes, you can. And you would just name them differently. So for example, let me see. So for example here, right? So this is the variable iris. And you can also have another variable called, I don't know, just flowers equals, you just like flowers, and then you would read in that file. And then whenever you want to manipulate it, you would call that specific variable that you store the data in. And so how do you foresee the future of data analysis in scientific research evolving, and what trends should researchers be aware of? So I think, I feel like now every scientist, or it's really encouraged that everyone should know how to code, simply because a lot of data sets are getting bigger and bigger. And it's simply not feasible to use more well, it's not feasible, but it will be much more efficient, and you can also collaborate much easier with other people. And so I would say, like, people would start coding more, like especially if you want to do more analysis, right? More complex analysis, or you want to automate things, right? You can set up pipelines to process your data. And all these things require you to know how to code. And trends that you should be aware of, it hard to say, also depends on your field, right? So for example, it really depends on your research question, and also like what, I think different fields, they have their own curve in this. And also, so my role here is just to share with you, basics of how to get started. because I didn't come from computer science background or coding background. Like I also started learning on my own during my PhD, and I think definitely you need to be aware of the way you process and clean the data, right? It's very easy to overlook these things, and also how can you make data sets compatible? So there was actually a Nature paper, I think back in 2020, where they have different people from the same group, or like different people analyzing the same data set. And they actually get very different results. So what does this mean, right? It really depends. You need to document everything, right? I mean, in research in general, you should document everything. You should document how your data is processed, cleaned, and also what kind of like how you process the data, right? What kind of function you used and why you use these variables, and why did you use these parameters? How do you specify these parameters? And I think these are things that you need to pay attention to and also document, and also make sure to comment in your code, especially when you collaborate with other people, right? So people know that what this line in your code is actually doing. So these are some, I guess, good practices to start with, to comment, right? It doesn't hurt to comment more, in my opinion, because it saves you in the future, right? You can go back and see why you did this at that point. So I hope that answers your question, and I hope that you all learned something today, and definitely check out the resources I recommended. And it really depends on it really depends on like what you're trying to do, right? So I think I know like a lot of people who do like single cell and data analysis, they use R. And then for Python, it's again, like I said, it's a more general purpose language. So, but I find Python easier to learn. So it's a very personal thing as well. So I hope you can utilize the resources that I shared earlier. And yeah, if you have questions, definitely reach out. I think you can find my profile there. And I'm very happy that I got to share my experience with you here today. And I wish you all luck in your research and your future endeavors.